In the last video, we discussed the overview of how energy is obtained from food and captured in ATP to fuel the functions of our bodies. In this video, we will dive into more details about each of the metabolic pathways and how carbohydrates, fats, and amino acids are broken down. As mentioned in the previous video, carbohydrate, fat, and protein are digested into glucose, fatty acids, and amino acids to be absorbed and transported through the bloodstream and into our cells. Each of these nutrients enters cells in different ways. Once inside the cell, they will each be broken down in separate parts of the metabolic pathways to obtain energy. Most of the initial breakdown of the macronutrients occurs in the cytosol of the cell. Eventually, the processes enter the mitochondria to produce ATP that will capture the energy extracted from carbs, fats, and protein. This is a large overview of the metabolic processes described in the previous slide. As mentioned, once carbohydrates, fats, and proteins enter the cell, they proceed to different parts of the pathway. Carbohydrates' initial breakdown is through glycolysis. Fat is broken down through fatty acid oxidation, and amino acids enter separate amino acid pathways. Let's take a look at each of these individual pathways, starting with carbohydrates, identified in blue at the top of the graphic. The initial breakdown of glucose is called glycolysis. Glycolysis occurs in the cytosol of the cell and requires no oxygen. The first thing to notice is the arrow between glucose and pyruvate is reversible. This is a very important concept because it means that at any point along this pathway, the process can be reversed and glucose can be made. Glucose is a 6-carbon chain, so it will actually be broken down into 2 pyruvate molecules. As the glucose is ripped apart through several reactions, hydrogen atoms with their high-energy electrons are released. A coenzyme made of the B vitamin niacin grabs onto the electrons and carries them to the electron transport chain. Looking back to the large overview picture, we can add the next step after glycolysis. Remember, we left off with 2 pyruvate molecules. Let's take a closer look at the options pyruvate has. This picture is slightly different than what we looked at on the previous slide, but is really just a simplified version of it. The box in blue is glycolysis. The steps we just went over breaking down glucose into pyruvate. Pyruvate has a couple of options. If cells require energy, pyruvate needs to be made into acetyl CoA, but this process of making acetyl CoA occurs in the mitochondria of the cell. Remember, glycolysis was in the cytosol. The conversion to acetyl-CoA also requires oxygen. In absence of enough mitochondria and/or oxygen, acetyl-CoA cannot be made. This happens frequently in muscle cells that are working at a very fast pace. One option cells have is to make lactic acid. It is a much faster source of energy, such as what one might need to sprint a quarter of a mile. In this case, muscle cells will convert pyruvate into lactic acid. Unfortunately, this is a short-term solution that cannot be sustained for very long. The best option is to make acetyl-CoA for a more sustained level of energy. But it's nice to have a backup plan. Let's switch gears away from the carbohydrates and move into fat, identified in the peach boxes to the upper right side of the graphic. If you recall from the beginning of the semester, 95% of the fats we consume are triglycerides. The triglyceride structure includes a glycerol backbone with 3 fatty acid chains attached. It is important to note the structure because these 2 different parts are broken down in different ways. The glycerol backbone is a 3-carbon molecule, much like pyruvate, so it can be easily converted into pyruvate. But only the glycerol unit can be made into pyruvate. Fatty acid chains cannot. In the breakdown of fatty acid chains, 2 carbons are cleaved off and coenzyme A is added to create acetyl-CoA. Of course, the hydrogen with its electrons are released. Coenzymes will carry these electrons to the electron transport chain to make ATP. This process continues, cleaving 2 carbons and adding a CoA until the fatty acid chain has been completely broken down into acetyl-CoA molecules. Now here's a question for you. Can fatty acids be made into glucose? Follow the pathway. 2 carbons, add a coenzyme A to make acetyl-CoA. Can acetyl-CoA make glucose? No. acetyl-CoA's arrow does not reverse to make glucose. The glycerol backbone of a triglyceride can make glucose, but not the fatty acids. We talked about this at the beginning of the semester. Fatty acids cannot make glucose. So let's go back to the big picture and point out what we just talked about. Glucose enters glycolysis, where it is broken down into pyruvate molecules. Pyruvate is then converted into acetyl-CoA in the mitochondria of the cell. With the triglycerides, the glycerol backbone will be converted into pyruvate, just as glucose was. The fatty acid chains are broken down 2 carbons at a time to make acetyl-CoA. Now, let's shift gears to amino acid breakdown. Different amino acids enter the metabolic cycles at various points. Some amino acids enter glycolysis, some are converted to acetyl-CoA, and others enter the Krebs cycle. The amino acids that enter glycolysis are called glucogenic amino acids. That should make a lot of sense because we talked about the fact that glycolysis has a reversible arrow. This means that the amino acids that are converted to pyruvate can also make glucose. The word glucogenic means glucose forming. Some cells require glucose exclusively, so this is an important fact because in the absence of glucose, amino acids will be converted into glucose. The amino acids that enter the Krebs cycle or are converted to acetyl-CoA are called ketogenic. And one important point to note is that these amino acids must go through a chemical change before they can enter any of the pathways. In order for amino acids to provide energy like carbohydrates and fat, the amino acids must chemically be the same as well. So before entering the pathways, amino acids must be stripped of nitrogen in a process called deamination. The end product of removing the nitrogen and hydrogens is ammonia. And this ammonia must be excreted. The process includes sending the ammonia to the liver, where it is converted into urea. The urea is then sent via the bloodstream to the kidneys, which will add the urea to urine for excretion from the body. Looking back to the big picture again, let's summarize what we have discussed so far. Glucose enters glycolysis, where it is broken down into pyruvate molecules. Pyruvate is then converted into acetyl-CoA. With the triglycerides, the glycerol backbone will be converted into pyruvate. The fatty acid chains are broken down 2 carbons at a time to make acetyl-CoA. Amino acids enter the pathway in various spots, but are largely converted into pyruvate or acetyl-CoA after going through deamination. If you notice, acetyl-CoA is at the center of all 3 macronutrients. All of carbohydrates, fats, and protein make acetyl-CoA. So if acetyl-CoA is at the center of the energy nutrients, the decision of the next step becomes critical. This is a simplified version of the picture on the last slide. We have glucose in blue, triglycerides in the peach color, and amino acids in pink. Looking at the arrows from acetyl-CoA, it appears we have 2 options. 1, acetyl-CoA can enter the Krebs cycle, or 2, acetyl-CoA can make fatty acids. The question is, what dictates which direction this goes? Well, if your cells require energy, then it is critical to continue through the pathway to Krebs cycle and onto the electron transport chain to make the maximum amount of ATP. So if energy is needed in cells, then acetyl-CoA enters Krebs cycle. However, if enough ATP has already been produced to sustain energy necessary for cells, then acetyl-CoA will take the remaining energy and make fatty acids to store in adipose cells. Does acetyl-CoA say, hey, maybe he won't be eating for another hour, so maybe I should wait to see if I should make fat? No. This process is in real time, meaning at that moment, if energy demand has been met, the remaining energy will be stored as fat. So if acetyl-CoA makes fatty acids, and acetyl-CoA is made by all 3 energy nutrients, you can clearly see the path of how all energy nutrients in excess make fat. This is another concept we learned early in the semester that is now clearly explained by the metabolic processes. I know this was a lot to cover. I will save the rest of this exciting saga for another video.